We live in a remarkable time, the age of genomics. Your genome is the entire sequence of your DNA. Your sequence and mine are slightly different. That's why we look different. I've got brown eyes, you might have blue or gray. But it's not just skin deep. The headlines tell us that genes can give us scary diseases, maybe even shape our personality or give us mental disorders. Our genes seem to have awesome power over our destinies. And yet, I would like to think that I am more than my genes. What do you guys think? Are you more than your genes? Yes? I think some people agree. I think some people agree with me. I think we should make a statement. I think we should say it all together. All right? I am more than my genes all together. I am more than my genes. What am I? I am my connectome. And since you guys are really great, maybe you could humor me and say this all together too? Right? All together now. I am my connectome. Oof, that sounded great. You know, you guys are so great. You don't even know what a connectome is, and you're willing to play along with me. I could just go home now. Well, so far, only one connectome is known, that of this tiny worm. Its modest nervous system consists of just 300 neurons. And in the 1970s and 80s, a team of scientists mapped all 7,000 connections between the neurons. In this diagram, every node is a neuron, and every line is a connection. This is the connectome of the worm C. elegans. Your connectome is far more complex than this, because your brain contains 100 billion neurons and 10,000 times as many connections. There's a diagram like this for your brain, but there's no way it would fit on this slide. Your connectome contains 1 million times more connections than your genome has letters. That's a lot of information. What's in that information? We don't know for sure, but there are theories. Since the 19th century, neuroscientists have speculated that maybe your memories, the information that makes you you, maybe your memories are stored in the connections between your brain's neurons. And perhaps other aspects of your personal identity, maybe your personality and your intellect, maybe they're also encoded in the connections between your neurons. And so now you can see why I proposed this hypothesis, I am my connectome. I didn't ask you to chant it because it's true. I just want you to remember it. And in fact, we don't know if this hypothesis is correct because we have never had technologies powerful enough to test it. Finding that worm connectome took over a dozen years of tedious labor. And to find the connectomes of brains more like our own, we need more sophisticated technologies that are automated, that will speed up the process of finding connectomes. And in the next few minutes, I'll tell you about some of these technologies which are currently under development in my lab and the labs of my collaborators. Now, you've probably seen pictures of neurons before. You can recognize them instantly by their fantastic shapes. They extend long and delicate branches, and in short, they look like trees. But this is just a single neuron. In order to find connectomes, we have to see all the neurons at the same time. So let's meet Bobby Kasturi, who works in the laboratory of Jeff Lichtman at Harvard University. Bobby is holding fantastically thin slices of a mouse brain, and we're zooming in by a factor of 100,000 times to obtain the resolution so that we can see the branches of neurons all at the same time. Except you still may not really recognize them, and that's because we have to work in 3 dimensions. If we take many images of many slices of the brain and stack them up, we get a three-dimensional image. And still you may not see the branches, so we start at the top, and we color in the cross-section of one branch in red. And we do that for the next slice, and for the next slice. And we keep on doing that, slice after slice. If we continue through the entire stack, we can reconstruct the three-dimensional shape of a small fragment of a branch of a neuron. And we can do that for another neuron in green. And you can see that the green neuron touches the red neuron at two locations. And these are what are called synapses. Let's zoom in on one synapse and keep your eyes on the interior of the green neuron. You should see small circles. These are called vesicles. They contain a molecule known as a neurotransmitter. And so when the green neuron wants to communicate, wants to send a message to the red neuron, it spits out neurotransmitter. At the synapse, the two neurons are said to be connected, like two friends talking on the telephone. So you've seen how to find a synapse. How can we find an entire connectome? Well, we take this three-dimensional stack of images and treat it as a gigantic three-dimensional coloring book. We color every neuron in a different color, and then we look through all of the images, find the synapses, and note the colors of the two neurons involved in each synapse. If we can do that throughout all the images, we could find a connectome. Now, at this point, you've learned the basics of neurons and synapses, and so I think we're ready to tackle one of the most important questions in neuroscience. How are the brains of men and women different? According to this self-help book, guys' brains are like waffles. They keep their lives compartmentalized in boxes. Girls' brains are like spaghetti. Everything in their life is connected to everything else. You guys are laughing, but you know, this book changed my life. But seriously, what's wrong with this? You already know enough to tell me. What's wrong with this statement? It doesn't matter whether you're a guy or girl. Everyone's brains are like spaghetti. Or maybe really, really fine capellini with branches. Just as one strand of spaghetti contacts many other strands on your plate, one neuron touches many other neurons through their entangled branches. One neuron can be connected to so many other neurons because there can be synapses at these points of contact. By now, you might have sort of lost perspective on how large this cube of brain tissue actually is. And so let's do a series of comparisons to show you. I'll assure you, this is very tiny. It's just 6 microns on a side. So here's how it stacks up against an entire neuron. And you can tell that really only the smallest fragments of branches are contained inside this cube. And a neuron, well, that's smaller than a brain. And that's just a mouse brain. It's a lot smaller than a human brain. So when I show my friends this, sometimes they've told me, you know, Sebastian, you should just give up. Neuroscience is hopeless. Because if you look at a brain with your naked eye, you don't really see how complex it is. But when you use a microscope, finally the hidden complexity is revealed. In the 17th century, the mathematician and philosopher Blaise Pascal wrote of his dread of the infinite. His feeling of insignificance at contemplating the vast reaches of outer space. And as a scientist, I'm not supposed to talk about my feelings. Too much information, Professor. But may I? I feel curiosity, and I feel wonder, but at times I have also felt despair. Why did I choose to study this organ that is so awesome in its complexity that it might well be infinite? It's absurd. How could we even dare to think that we might ever understand this? And yet I persist in this quixotic endeavor, and indeed, these days I harbor new hopes. Someday, a fleet of microscopes will capture every neuron and every synapse in a vast database of images. And someday, artificially intelligent supercomputers will analyze the images without human assistance to summarize them in a connectome. I do not know, but I hope that I will live to see that day. Because finding an entire human connectome is one of the greatest technological challenges of all time. It will take the work of generations to succeed. At the present time, my collaborators and I, what we're aiming for is much more modest, just to find partial connectomes of tiny chunks of mouse and human brain. But even that will be enough for the first test of this hypothesis that I am my connectome. For now, let me try to convince you of the plausibility of this hypothesis, that it's actually worth taking seriously. As you grow during childhood and age during adulthood, your personal identity changes slowly. Likewise, every connectome changes over time. What kinds of changes happen? Well, neurons, like trees, can grow new branches, and they can lose old ones. Synapses can be created, and they can be eliminated. And synapses can grow larger, and they can grow smaller. Second question, what causes these changes? Well, it's true. To some extent, they are programmed by your genes, but that's not the whole story, because there are signals, electrical signals that travel along the branches of neurons and chemical signals that jump across from branch to branch. These signals are called neural activity. And there's a lot of evidence that neural activity is encoding our thoughts, feelings, and perceptions, our mental experiences. And there's a lot of evidence that neural activity can cause your connections to change. And if you put those two facts together, it means that your experiences can change your connectome. And that's why every connectome is unique, even those of genetically identical twins. The connectome is where nature meets nurture. And it might be true that just the mere act of thinking can change your connectome, an idea that you may find empowering. What's in this picture? A cool and refreshing stream of water, you say. What else is in this picture? Do not forget that groove in the earth called the stream bed. Without it, the water would not know in which direction to flow. And with this stream, I would like to propose a metaphor for the relationship between neural activity and connectivity. Neural activity is constantly changing. It's like the water of the stream. It never sits still. The connections of the brain's neural network determine the pathways along which neural activity flows. And so the connectome is like the bed of the stream. But the metaphor is richer than that, because it's true that the stream bed guides the flow of the water. But over long time scales, the water also reshapes the bed of the stream. And as I told you just now, neural activity can change the connectome. And if you'll allow me to ascend to metaphorical heights, I will remind you that neural activity is the physical basis, or so neuroscientists think, of thoughts, feelings, and perceptions. And so we might even speak of the stream of consciousness. Neural activity is its water, and the connectome is its bed. So let's return from the heights of metaphor and return to science. Suppose our technologies for finding connectomes actually work. How will we go about testing the hypothesis, I am my connectome? Well, I propose a direct test. Let us attempt to read out memories from connectomes. Consider the memory of long temporal sequences of movements, like a pianist playing a Beethoven sonata. According to a theory that dates back to the 19th century, such memories are stored as chains of synaptic connections inside your brain. Because if the first neurons in the chain are activated, through their synapses they send messages to the second neurons, which are activated, and so on down the line, like a chain of falling dominoes. And this sequence of neural activation is hypothesized to be the neural basis of those sequence of movements. So one way of trying to test the theory is to look for such chains inside connectomes. But it won't be easy because they're not going to look like this. They're going to be scrambled up. And so we'll have to use our computers to try to unscramble the chain. And if we can do that, the sequence of the neurons we recover from that unscrambling will be a prediction of the pattern of neural activity that is replayed in the brain during memory recall. And if that were successful, that would be the first example of reading a memory from a connectome. What a mess. Have you ever tried to wire up a system as complex as this? I hope not. But if you have, you know it's very easy to make a mistake. The branches of neurons are like the wires of the brain. Can anyone guess what's the total length of wires in your brain? I'll give you a hint, it's a big number. I estimate millions of miles, all packed in your skull. And if you appreciate that number, you can easily see there is huge potential for miswiring of the brain. And indeed, the popular press loves headlines like anorexic brains are wired differently or autistic brains are wired differently. These are plausible claims, but in truth, we can't see the brain's wiring clearly enough to tell if these are really true. And so the technologies for seeing connectomes will allow us to finally read miswiring of the brain, to see mental disorders in connectomes. Sometimes, the best way to test a hypothesis is to consider its most extreme implication. Philosophers know this game very well. If you believe that I am my connectome, I think you must also accept the idea that death is the destruction of your connectome. I mention this because there are prophets today who claim that technology will fundamentally alter the human condition and perhaps even transform the human species. One of their most cherished dreams is to cheat death by that practice known as cryonics. If you pay $100,000, you can arrange to have your body frozen after death and stored in liquid nitrogen in one of these tanks in an Arizona warehouse, awaiting a future civilization that is advanced enough to resurrect you. Should we ridicule the modern seekers of immortality, calling them fools, or will they someday chuckle over our graves? I don't know. I prefer to test their beliefs scientifically. I propose that we attempt to find a connectome of a frozen brain. We know that damage to the brain occurs after death and during freezing. The question is, has that damage erased the connectome? If it has, there is no way that any future civilization will be able to recover the memories of these frozen brains. Resurrection might succeed for the body, but not for the mind. On the other hand, if the connectome is still intact, we cannot ridicule the claims of cryonics so easily. I've described a quest that begins in the world of the very small and propels us to the world of the far future. Connectomes will mark a turning point in human history. As we evolved from our ape-like ancestors on the African savanna, what distinguished us was our larger brains. We have used our brains to fashion ever more amazing technologies. Eventually, these technologies will become so powerful that we will use them to know ourselves by deconstructing and reconstructing our own brains. I believe that this voyage of self-discovery is not just for scientists, but for all of us. And I'm grateful for the opportunity to share this voyage with you today. Thank you.
The precision of a watch is a function of its movement. For Rolex and for Hans Wilsdorf, to guarantee the precision of a timepiece, the pressing question was how to protect the movement itself from the elements, not only water, but also tiny particles of dust. In 1926, a major step was taken with the creation of the world's first waterproof and dustproof wristwatch. the Rolex Oyster was born. Over the years, subtle changes in the design continue to improve the Oyster, adding more comfort while keeping the style contemporary. And, along with style, more functions have been added. A Rolex wristwatch was the first to show the date through a small aperture on the face. It was also the first wristwatch to spell out the day of the week in full. In the early 1950s, Rolex developed professional watches whose functions went far beyond telling the time. Launched in 1953, the Submariner was the first Rolex watch guaranteed waterproof to a depth of 330 feet. Already on an incredible journey of innovation and design, Rolex decided to push the boundaries even further. In 1960, the bathyscaphe Trieste and Rolex made history. The submersible successfully dived to 35,800 feet below the surface of the ocean. A Rolex Deep Sea Special was strapped to the outside. The development of undersea exploration led to the launching in 1967 of the Sea Dweller 2000, waterproof to a depth of 2,000 feet. In 2008, the Submariner in gold is redesigned and the case features a new unidirectional rotatable bezel with a Cerachrom disc. Fitted with the patented Rolex ringlock system, the Rolex Deep Sea safely descends to 12,800 feet. Rolex has incorporated countless hours and more than a century of experience, years of research, innovation, and development into every one of its models. And the benefits arising from this work, including waterproofness, precision, and durability, are the result of Rolex's continuous pursuit of perfection. From the most elegant and prestigious models to the professional timepieces, all are exquisitely crafted. Piece by piece, we design and manufacture every single watch. And the story continues.